Welcome you all to this cryo-EM course, and this will be the last anatomy part of the microscope. So, we started from electron gun, then we discuss about the electromagnetic lens system, and then different component of this electromagnetic lens, starting from condenser lens to objective lens, then projector lens and intermediate lens. And now we discuss about the different pump system when you are collecting our data. Now we are going to discuss about the detector system, and that is one of the most important system for that electron microscope. There is a majorly 2 or 3 different types of detectors we are using, and why we want the different types of detector for the biological science sample, that is I am going to discuss today, and how it is going to help us when we will do our imaging, especially for biological science sample when we are using the cryo imaging at cryo temperature. Let us take us a look how these things are working. So, mostly for cryo-electron microscope we are using a different types of detector. So, first I want to show you a very basic detector system, that what kind of detector we can use it. So, here this is you are familiar with, this is the electron microscope, right, and this is the viewing chamber through where you can see all the samples, and this is the binocular. Now, you have a previously long back, if we just consider 10-15 years ago, we do not have any other different types of camera. So, that time CCD camera was not that great or that good, and most of the times we are using the plate camera for imaging purposes, and this is the plate camera. Sometime we are using the fluorescent screen, that is I am writing here fluorescent screen, then TV is attached with this microscope and we can see those things. So, in this microscope now computer is available, and that is the reason the computer is attached. But if you just consider that 20 years ago how we can get those images, and computer is not that easily available everywhere, right. So, here this is the chamber where we are keeping that photographic film. So, those are the photographic film, we are keeping it at the top. If you look at, this is the photographic film cassette, you can say, where we can put this photographic film inside, and when it will go inside. So, if you just look at, this will be the 6 centimeter and this will be the 9 centimeter, and this is the entire photographic film which is fitted in this particular cassette, and this individual cassette will go here, and we have to stack one by one like that. So, total 56 film you can stack here, and this 56 film entire cassette, it will go at the back inside the microscope. So, this plate camera we can fit it at the bottom here, and then this is the handle, it will bring that entire plate out here, and it will go through the exposer and it will store at the bottom. When this entire film it will stored at the bottom, we have to take out these things, go to the dark room and develop this film. Most of the time for the cryo imaging we are using a extremely low dose, and if you remember I told most of the time that we have to use only 40 to 80 electrons per angstrom square, and that is the reason we are using higher spot size and the intensity, fixed intensity and the aperture to reduce the electron dose. And when electron dose is very low, our this photographic film should be very sensitive to record the data. If it is not very sensitive, it will be very difficult. So, those days we are using SO 163 film, and then dark room we have to develop that film. When that film is developed, then we have to scan this one and get a digital image. So, this is the entire process how we are using that electron detector or camera, plate camera. You can consider that almost 15 years ago. But after that there is a lot of development, right. We are most of the time using charge coupled device, that is called the CCD camera, or nowadays we are all using the direct electron detector, CMOS kind of camera. So, let us discuss little bit more that what is the difference between those camera. So, here this is a one schematic diagram of a CMOS camera, CCD camera, where this is the scintillator coupled detector. So, here you have a scintillator where electron it will hit, it will convert to a photon, and then it will travel through a fiber optics cable, and when it will travel through this fiber optics cable, it will then this is the readout sensor where it will record those images. So, here you can see this is the CCD architecture where there is a pixel, different pixel, and electron is coming and interacting with this pixel, and it will go through this fiber optics coupling cable, and then there is a readout output sensor. So, there you can see the signal is recorded and we are getting that image. On the other hand, there is a direct detector. So, direct electron detector, basically there are three major direct electron detectors still available in the market. One is called the from the Gatan, and it is either K2 or K3 direct electron detector, and from the Thermo Fisher you have the Falcon 3 or Falcon 4, and direct detector is the another one that is possible. In all the cases it is detecting the electron directly. We do not need this fiber optics part at all, and you can see that electron when it hit to the sensor scintillator and it is converted, it is traveling a long distance. How it is happening in next slide I am going to explain, but in this particular case you can see this fiber optics cables are not there, it does not need to travel, it will hit to the scintillator and directly record. So, distance is small and does not need to travel, it is not generating that much noise, and it is very precisely recording, and that is the reason readout or output is very fast. So, this is the CMOS architecture. When it is coming, it does not need to travel, it is just transferring the signal in the on the chip processing, and we are getting the output image. So, the illustration of the scintillator coupled detector in which the incident electron are converted to photon. Here the incident electron first converted to the photon, and then photon is transferred and recorded into the sensor, which travel through a fiber optics coupling before reaching to the sensor. That is a drawback of the charge coupled device, where the illustration of the direct detector where electron are directly incident on a radiation and recorded the sensor. This is the two major difference between the direct detector and the normal charge coupled device. Now, how this charge coupled device is working? As I discussed, the charge coupled device, it has a scintillator. This is the P43 scintillator where electron will come, interact, and it will convert it to photon. So, here the fiber optics is there. When electron hits here and it is go through this fiber optics, and this is the pixel where it is sensor, where it is recording those photons to the sensor, which is giving the actual image. Now, when there is a scintillator, whatever electron and photon it will interact, it will generate some kind of signal which is travel through this one. One of the major problem is that here you can see there is a long distance it has to travel. When it is traveling through that, some of them are deviate in a different direction. It will be not all the time it is going to travel. So, most of the time some simulation data showed that 300 kV electron are crossing this barrier very fast, but when there is a 100 kilovolt electron or 200 kilovolt electron, they travel in a different direction. So, sometime they will go back and hit this scintillator again. When if electron is coming and going through this and it deviated from this path and hitting this scintillator, the scintillator will consider that this is another event, another single electron event, and that will generate a signal again, and that will travel in a different direction. Sometimes those kind of signal, it is generating a noise. So, if we look at that next diagram, we can see that how that electron is hitting the scintillator and traveling through that fiber optics. So, if we just go to the next, so what is the major difference between that between our charge coupled device and the CCD? The charge coupled device, the major difference is this, that it converts electron to a signal first. So, electron will be converted to the photon. Okay. Then it will start transferring that signal. So, it cannot directly transfer the electron. Then it will detect the signal with the sensor, and then electronically transfer this signal and read out to form image. So, there are 4 different steps are there, and couple of steps can create noise or other problems. So, what I was trying to explain in the last slide. So, sometime what has happened, that if electron is coming here, we are expecting that electron will go straight, but most of the time after hitting, that electron will just start spreading in this direction. So, it will be like all this direction. Now, based on that total charge of that if it is a 300 kV microscope or it is a 200 kV microscope, as I mentioned, they have a more energy, that 200 kV 300 kV. So, they can spread more, but if it is a 100 kV or 200 kV, the spreading will be less, and then they will follow different kind of trajectory. It will not follow all the time this straight line. So, if it will follow a different types of trajectory and couple of electrons, so it will go back and hit this, then what will happen? It will consider that this is another event, this is another single event, and this will start generating the electrons in all the direction. So, sometime they have a less energy of those electrons than these electrons, and that will create more noise. So, that is one of the reason the CCD camera definitely it is better user friendly than photographic film, because you can continuously collect many images, and your images are comparatively, you can see it immediately, you do not have to develop in a dark room. So, definitely that is user friendly, better resolution you will get it, but there is a problem that it will generating a lot of noise because of this kind of scattering, as well as it has to travel through this, it has to convert from electron to photon, then photon is going to travel through this fiber optics, and then it is going to record, and then it is amplify and give us an image. Lot of time these images will be noisy images, not a very good image, and the readout will be also very slow because it has to go through this entire process. So, here that is I am talking about that Monte Carlo simulation plot of the electron trajectory, which is calculated for P43 scintillator. So, if this is a 100 kV, so here different image, it mentioned that what kilovolt we are using. So, it is from 100 kV, this is from 200 kV, this is 300 and this is 400. Now, this is 100 kV, you can see electron hits the scintillator, even it is not traveling that fast, but in the other cases you can see a different kind of trajectory, they are following a different types of path, and this is basically calculated using this particular method, that Monte Carlo simulation. But when you have a more energy electron, they travel longer distance, they have a different type of trajectory. So, even in 400 volt you can see some of these electrons are going back here or going back here, and it is started again a new event. So, that is creating some kind of noise. Also, readout is very slow, that is one of the reason we can consider that it will always give us an integrated image. For any kind of imaging we need some kind of exposure, and this exposure time is very important. So, if exposure time something that 4 second, 5 second, 8 second, what it will happen, it will just give us an one integrated image. It cannot give a separate images. 4 second exposure it will take and one single image it will give. And if you remember clearly, when we discuss about that in cryo-electron microscope, what we are doing, basically we have a grid, and inside that grid we have a square. If we go inside that square, we will get a hole. There is a amorphous ice, and protein is embedded at different orientation. When electron beam will pass through, we will get 2D image. And when there is a amorphous ice and when electron beam is going through that sample, there is a lot of energy, and that will generate a thermal drift. At that drift, basically you will be in a continuous motion, and if we are talking about that we want a high resolution structure, that time this drift is going to affect our images. That is the reason we need a very fast readout. So, even if we are using 4 second exposure, we do not want to collect a one integrated image of 4 second. 4 second we want to divide by 10, and every 0.4 second I want to collect a single frame. So, in the later stage I can align them properly, so we can correct the drift. So, that is the reason it is very important to fast readout, which is not possible when we are using the CCD camera. That is only possible if we are directly collecting that electron and recording, it is hitting the scintillator and then recording that signal immediately. So, then readout will be fast and we can correct any kind of motion. So, when I will come to the real data, that time it will be clear. Let us take a look that what kind of differences are there when you are talking. So, this is the similar kind of simulation and trajectory of the electron at a different energy of that electron, and we can you can see that when there is a less energy, that the trajectory is not completed, but when there is a high energy, it is spreaded all over. Now, this is the difference, we can see it between a charge coupled device. This is the CCD camera, and this is, you can consider that direct electron detector, you can say direct electron detector. So, here the charge coupled device, what it is doing, as we discussed already, this is a scintillator, electron is interacting with the scintillator, then it converted it to photon travel through this optical fiber, and then there is a sensor, and from the sensor the recording is transferred and we are getting an image. But for the CMOS camera, you can see this is the sensor where the electron is directly interacting with the sensor and it is transferred that signal immediately and it is recording. So, there is no convert from electron to photon and then you have to record. Here the electron directly interact with the sensor and converted it to photon and the recording this photon in this sensor. So, now this we can split it into two half, that is the bulk direct detection and the back thin direct detection. So, that is telling us what is the thickness of the sensor. If the sensor is a smaller and it can recognize a good signal and it can transfer that signal easily to the readout, and also we do not have to travel this distance. So, readout will be much faster, as well as it will generate a less amount of noise. So, that is one of the good thing when you are using the direct electron detector. So, now in this one it will be much more clear how the signal is transferred. So, here you can see that the scintillator is here, which convert the electron to photon, it travel in a different direction. When there is a travel in a different direction, it is not recording directly. So, the chances of noise is much higher, and then we are going through that light transfer, then it is recording, and then we are getting the readout. The other case, it has to travel, that we mentioned couple of times, and that is generating noise also. Where the direct electron detector, this is the sensor, it is hitting the sensor, and directly electron it is taking care. We do not need to convert from electron to photon anymore. So, this requirement is not there when you are using the CMOS camera or direct electron detector. So, that is one of the good thing. So, now when we have that electron recorded hit the sensor, immediately it will record that all the signals to the readout, and we are getting a much faster images, much better images. So, that is going to help us for our biological science sample. As we all know that biological science sample is temperature sensitive, as well as this cryo sample is staying inside the amorphous ice. That is the reason direct electron detector is very important for our purposes. So, here couple of points are important points are written here. What it is telling, there is a here are the only two steps in TEM imaging with the direct detection, where the CCD camera there are 4 steps: hitting the scintillator, then convert it to photon, then photon will travel through the optical fibers, and then it will record, and then we will get the readout. That is for the CCD camera. But for the direct electron detector, convert from electron to signal not applicable, it is not required. So, detect direct electron, that is one of the good thing. Then transfer signal, again another important point, where the CCD camera the transfer signal is happening from the sensor to readout. Here this one is not applicable, no need to transfer the photon signal to fiber optics. So, that part it is good thing. Detect signal with sensor directly. So, electronically transfer signal and read out to form the image. So, these are the 4 important points that is helping us to fast read out and get very good quality and high resolution images. And also because of this fast readout, which is helping us to correct the drift, and it is preserve most of the high frequency signal. High frequency signal when we are preserving, that will help to get a high resolution structure. Now, those high frequency signal, low frequency signal, that is we are going to discuss in the next class. Here what we have to understand, that why we are suddenly interested about the high frequency signal, recording all of this signal. These high frequency signals are very important when we are trying to collect a high resolution data. High resolution data means when we want to see a protein structure, we want to see the helix of a protein, or DNA helix, or RNA secondary structure, tertiary structure. We want to see the beta strand, we want to see the side chain of the amino acids. That means that we have to see the tyrosine, tryptophan, lysine, glutamic acid side chain. That is the time if there is a thermal drift. So, if this is a sample we want to image, and this is even a micrometer, if you just consider that 0.1 micrometer, 0.2 micrometer movement is there because of that, because of that electron, and there is a little bit drift 0.1 every second, that drift is also huge when we are considering to see the side chain of a protein or side chain of an amino acid. If we want to just image a bacterial cell, probably we do not need that kind of high quality image, high quality detector, because bacterial cell we are not looking at the membrane, how those outer membrane is formed. If we just want to image a bacterial cell or normal cell, we want to do a room temperature TEM, we do not need direct electron detector. So, in this particular case, we have to decide at what particular detector it is required for me. Why? For the negative stain or room temperature TEM we do not need direct electron detector, because room temperature TEM what we are using, we have a grid square. If we just consider, as I mentioned previously, this is our grids, right? You are looking at that grid where you have a carbon layer at the top all over. It will be a carbon layer. So there will be no whole kind of thing that where our protein sample will go. This is only required for cryo. So we can erase this diagram because that is whole concept is not there anymore. What is required here? What is required here, that it is a normal room temperature temp, and this room temperature temp we have that grid, and we are looking at the one square of the grid which is coated with amorphous carbon, and then we are adding our sample on top of that. We will discuss about this one when we will discuss about the sample preparation. Here we are just discussing why we do not need direct electron detector for room temperature temp. So, when we are adding sample, a excess sample will be there on top of that. So, this will be the square where our buffer sample and buffer as well as the protein sample will be all over. Then we will use a blot paper. This is our blot paper. Then we will blot the excess buffer. Then we will use uranyl acetate or uranyl format to stain this grid, and we will keep it at dry condition at room temperature. Keep the entire thing at room temperature for 10-15 minutes. When this grid will dry, we take this one and image it. Lot of time we keep it inside the desiccator also to keep it dry. So, when our sample is already dried, means almost 70 to 80% water is removed from the sample. That means the sample is already collapsed. We are not thinking about to see any protein structure, that helix or beta strand, hydrogen bond, hydrophobic interaction or electrostatic interaction. So, for negative staining it does not matter, as well as sample is not staying inside a hole where you have a amorphous ice. In the cryo condition you have a ice, and this is the carbon area as I discussed, and inside the ice you have the sample, and you want to image. If you have a amorphous ice and electron beam is passing through this sample, the high chance there will be a thermal drift or there will be a motion, continuous motion. That motion is not possible when you are imaging something in a dry condition and when you have a carbon platform and sample is not in amorphous ice. So, in this particular cases you are not targeting a very high resolution, means 1 angstrom, 2 angstrom or 3 angstrom structure, and you are not expecting that you can see the side view from this one. You just want to see the overall architecture of the sample, overall architecture of the protein, overall architecture of the bacteria, overall architecture of the cell. You want to see the mitochondria. You are not targeting that what is the arrangement of the lipid, what is the arrangement of the any protein which is expressed in the mitochondria or in the bacterial cell. You are not looking for a secretion system directly. So, in these cases, even if you have a camera like a CCD camera, it does not matter, or a low quality CMOS camera is good enough for getting a high resolution data using the normal room temperature temp. So, definitely for room temperature temp we do not need a direct electron detector at this moment. Now, for the cryo, definitely as I mentioned couple of times, we need to collect data very fast. So, we can correct the drift, and that is one of the reason we want a good quality direct electron detector which is helping us to collect a high resolution data when you will image any biological science sample. Now, let us go to the next one. So, we already discussed about the readout thing, and we already discussed how this is affecting to the scintillator. If this is a scintillator as I mentioned for the CCD camera, electron hits and then electron is traveling through the fiber optics, and some of this electron will travel in a different direction. If it will hit, it will generate again some signal. So, here you can see this is collected from a CCD camera. In the CCD camera you can see Some of these spots are very bright spot, some of the spots are very light spot. If we just look, so here you can see that those spots contrast is very low, but some of the spots contrast is very big, and the spot size is also very different size. Now when the electrons are traveling in a different direction like that, and if they hit, they will generate more electrons, and everything is going to record here, and that is basically going to affect this kind of things, that even there is a single electron event, it will just not uniformly distributed. Some area it is distributed very strong intensity, and some area you can see distribution is very less. But when you have a direct electron detector, something like that, you can see that there is a bright spot here, but most of the bright spots are very uniformly distributed, because the signal, it is, there is a sensor and immediately there is the readout. So there is no need to convert from electron to photon, and then you have to record, and that is the good thing for the direct electron detector, and that is one of the reason we are getting a very uniform distribution for the direct electron detector also. Now here is, for any camera, this term is very important, detective quantum efficiency. What is the detective quantum efficiency? It is depending on the signal to noise ratio. So here you can see high signal to noise ratio is good, because if signal to noise ratio is almost equivalent, so if signal and noise is almost close side by side, it is, your images will be not that great, right. So if we can increase the signal to noise ratio, means signal is much higher, that will be good, and that is called the detective quantum efficiency. So here the mathematically DQ is expressed as the relationship between the output and input of the signal to noise ratio. So the signal to noise ratio out squared divided by signal to noise ratio in squared. So that is called the DQ. If DQ is higher, your image quality will be much higher, your contrast will be much better. So here I am showing you a image. So this is the definition of the detective quantum efficiency. So measure how efficiently an imaging system converts the incident photon or electrons into the detectable signal, with a higher DQE indicating the better image quality for a given dose, because dose is always limited, that we have to keep it in mind. Now I am showing you an image. Any of you can tell me what image it is. It is just a question, you can think about that. So here I am using a image where, as I mentioned that almost signal to noise ratio is equivalent. It is, there is some images there, we do not know what image it is. So it is looking like that something is spreaded here, something is spreaded here, and something is spreaded here. But in this particular image, if you look carefully, here some signal is there, but noise level is so high. So in this formula, if you look at this formula, this SNR, that SNR in and SNR out. So definitely SNR out and SNR in is almost equivalent, or it is some sort of SNR out is lower, and that is the reason we cannot see the proper signal. But if we gradually increase the signal, readout signal, that SNR out is much higher, so DQ will be much higher. So now you can see it is much more prominent, it is looking like that some kind of, it is some kind of architecture, it is like this, and something else is also going on there, but still it is not clear. So in this case we can consider that almost signal to noise ratio, you can say 1:1. It is not that prominent, that noise level is so high, the signal is just equivalent to noise. So we cannot figure it out what it is. But if you just reduce the signal, if you just have a good quality of camera and we can record a very good image, you can see this is the image. So you can consider that this is my own image, I take this one and then at a different noise level, I am showing you what kind of differences you can see it. So, in the same image you can see it that when signal is good, if signal is not good, if there is too much noise, this image will be equivalent to this particular image. Okay. But if signal is good, you can figure it out that okay, definitely this is a human being and this is the background image it is coming. So, here the output image, the image after recording with a camera with a uniform 33% DQE. So, we will discuss about all of this image formation and how these noisy images we are getting in the TEM, those kind of noisy images can, if we use a model that can give us a biased model reconstruction, that we will discuss more. But here that is the take home message for today's class, that if signal to noise ratio is high, and that is very much required to get a good contrast, good images which will help us in the future for the 3D reconstruction. We will discuss about this imaging part in the next class. Thank you all for your attention.